Hello everyone, I am Dr. Devi, Associate Professor, Department of Genetic Engineering, SRM Institute of Science and Technology. I am here today to explain and demonstrate another important experiment which is part of the Animal Biotechnology Practical, thawing and revival of cell lines. As we all know, cells have to be cryopreserved so that they can be used whenever required for research purposes. Thawing is a process in which the frozen cells, which are stored either at minus 80 degrees centigrade or at minus 196 degrees centigrade in liquid nitrogen, are brought back to room temperature and converted from a metabolically inactive state to an active state. During this process, we have to ensure that there is minimal cell damage to make sure that there is maximal cell revival. Maximum cell revival can enable scientists and researchers to carry on their research and to continuously passage these cells. Thawing is a stressful process to the cells and therefore it has to be done really fast. It is ideally better to store the cryovials in the vapor phase of the liquid nitrogen tank. Also, make sure that you use culture media which has been properly sterilized. During the thawing process, the cryovial should be placed inside the water bath at 37 degrees centigrade by holding the lid of the cryovial. It should not be completely submerged into the water bath because this can increase the risk of contamination. The centrifugation RPM and time vary depending upon the cell type which you are using. After centrifugation, you can observe the pellet of cells, cryopreserved cells at the bottom of the centrifuge tube. Now take it back again to the biosafety cabinet, spray it with 70% ethanol, wipe it dry and open it inside the biosafety cabinet. Remove the supernatant and make sure that it is discarded. The supernatant is discarded because it contains the cryoprotectant DMSO or the dimethyl sulfoxide. After this, resuspend the pellet with required volume of culture media and transfer this to a culture flask. It can be either a Petri plate or a T25 flask and add required amount of culture medium. Shake it gently and place it inside the incubator. allow the cells to adhere, and after 24 hours you can observe the cells again to see if there is any cell debris or if there is any cell death. If there is high amount of cell debris, or if you can see a number of cells which are dead, you have to remove it and replace it with fresh culture media. If after the thawing process you observe that the cells are not growing properly, or if the cell yield is poor, it could be because of many reasons. It could be that the stocks were not stored properly, or the frozen conditions were not correct.